Okay, next speaker is Damla. Well, we've already heard from her. But she's going to talk about the work she's been doing at Bionano Genomics in this talk. Just so if there are people joining now, Damla was a PhD student in my group at CMU. She was the last student to graduate at CMU, from CMU, let's say. And she already presented the work that she had done, a part of the work that she had done for her PhD thesis. And then after that, she joined Bionano Genomics as a staff software engineer in hardware acceleration, as you can see. It's kind of an interesting, let's say, title. Software engineering and hardware acceleration group. But we're going to learn more about that right now, I guess. Damla, please go ahead.
Hello again. So this will be a different taste of presentation now. And as Onur covered, this will cover what I'm doing right now at Bionano. And in Bionano, our goal is, the vision is transforming how the world sees the genome. And the reason is that we are hearing a lot about sequencing, right? And what we are doing at Bionano is not sequencing, it's a different type of genome analysis. And that's how we are wanting to transform. So, and let's start. So this is our genome variant continuum, if you will. And here we are seeing on the left-hand side, starting from a whole chromosome up until one base pair. It's a continuum, as you can see it. And these are the coloring is not great, but I hope it's visible. So, and we are dealing with a lot of type of genomic variants out there. And as you can see from the spectrum, there is no single technology, if you will, that can address all of it. So on the left-hand side, we are looking at karyotyping or FISH or arrays to look at more chromosome level changes or variations that we can observe. And this part is called cytogenetics because it is looking at the chromosome level. And on the right-hand side, which we are extremely familiar with, we have sequencing. But as you can see from the spectrum, its range is pretty limited. So, and when we look at the cytogenetics, all of these almost 50 years old operations, the techniques that's out there have their own limitations. For example, FISH is targeted that you can only look at one variant, one type of variant or in one location. Or in arrays, you are only able, so this, the resolution is high. As you can see, it's spinning a lot, but it can only look at gains or losses, more GUS kind of analysis. And when you look at karyotyping, it's extremely labor intensive. And people really don't want to do it anymore. And it's very, as I said, there's not really automated system as we are observing in, for example, sequencing. So as the title mentions, there's a gap between and to fulfill the full, the complete of this genome continuum, because in order to reveal the answers, we need to be able to observe or reveal all types of genomic variants, which can start from one BP like a SNP, or small indels, or large structural variants. And in order to close that gap, to serve that unmet need, we are hearing a lot of improvements happening in the sequencing world for almost 2 decades. And recently, not recently, but all of these companies are introducing either improved machines or newer machines in order to, as I said, fill that gap. But let me go back. We can see in the long read, even in the long read world, we are pretty limited in terms of the continuum. And another issue is, again, highlighted here, the length of the read is a problem. So even if we have perfect sequencing with less like errors and those kind of things, there's this amount of gap available still need to be addressed with efficient workflows to be able to answer all the questions. And we are not there yet. And we, in genomics or bioinformatics, we love analogies. So I will introduce a new one here, and that will motivate us why optical genome mapping is needed. So this is, think about it like a book, and all the papers are shredded, and we don't know this, we don't have any idea about what this is about, and we have all these words. So when we look at just the words, we can like catch some of those spelling errors, like the synapse, you can think about them, or some indels maybe. But for example, we have this word, we have no idea without knowing the context, whether it is desert, or if it is desert, is it a verb, is it a noun? So we have no idea without having any idea about the context. So that's what optical genome mapping that Bionano is focusing on is proposing here. It's providing a different perspective. It is providing a different angle view to the genome. It's providing the context, basically. It doesn't have the resolution of sequencing. We cannot go as low as one base pair level, but we are looking at the chromosome, maybe a full arm of chromosome in an automated way. So we have an end-to-end workflow to do that. That's what optical genome mapping, or OGM, is putting to the table. And as you can see here, what we believe is OGM with Bionano is closing that gap. So it is covering all the cytogenetics approaches that's out there, which is a status quo basically that we are trying to replace. And also it's covering the missing piece from the sequencing world as well. And as you can understand from my title as well, the one of the goals is transforming the genomic analysis as well. But as you can see from here, the approach we are following is OGM has this missing piece as well. So OGM plus NGS, like short read sequencing, is the way to perform the, transform how genomic analysis is being performed. And in order to do that, in the last several years, within the company, there are several acquisitions happening. For example, I will match this figure with one of my figures from my first talk. So if you remember, we are starting with a sample prep, and we go through a wet lab operation, and then we get the DNA, which is exactly similar, not exactly same, but similar enough in OGM workflow as well. And afterwards, you can put that, of course, how you prepare the DNA is, or the sample differs between technologies. They have their own techniques. But from a sample, you can gather something that is suitable for OGM, like our Sapphire system, and a sequencing. And one of the acquisitions was for the initial wet lab part, for example. And another acquisition was for the last part, data analysis and ex-clinical from biodiscovery. So our goal is kind of like filling the cytogenetics gap with OGM and coupling it with NGS so that we can have an end-to-end workflow that covers the missing pieces from each. And as I said, the important thing is revealing all types of variants, not just several types of that. So I will go over the OGM workflow to give you an idea, especially the last part, which is of interest to the group, the analysis part. But some details are important in the initial parts as well, so I will go through end to end. Starting with isolating the ultra-high molecular weight DNA, the next step is labeling it with and staining specific sequences. So there's a specific sequence with this, and we call it DLS. We stain the isolated DNA with that specific length, specific characters, and which means that when we label it within the DNA, whenever that sequence appears, we have a label out there. So as I mentioned, reminder, we don't look at the genome in the base pair level. We are looking at a higher resolution, like larger view. And after we label it, stain it, we transfer the labeled DNA into the chip, and we load this chip into the instrument. And you can think it like that. So this is how we label it, stain specific parts. And within the instrument, what's happening is that DNA is being linearized, and it's going through nanochannels to achieve that. And then the labels are being imaged. So we are taking snapshots, basically, and within repeated cycles. And then we gather these kind of images out of the instrument. And at that point, the analysis starts. And this is basically the primary analysis, if you would want to call it that way. What's happening is we get these images and convert them into molecules in Bionano vocabulary. And they are basically the digital maps, what we achieve. So they are basically saying in this relative position, so we are talking about positions and they are floating points, which is important to understand the complexity of the secondary analysis that we are dealing with. And we are getting those molecules out of these images, which contains the relative positional information out of the original genome, basically. And those molecules are the inputs to our analysis pipelines. And when we think about the pipeline, it's pretty similar in terms of major stages we are performing. But what we are dealing with, again, not ACGTs, but these locations, relative locations of these labels. And then what we do is we align, again, these molecules to reference maps. And then we construct a consensus genome map. And by using that and comparing it with reference maps, we are visualizing and revealing structural variations, basically. And this is one of our famous plots called circus plot. It's another Bionano vocabulary. So this is the end. Yeah, go ahead.
The locations represent what exactly? Is it location in the pore itself, the barcoding?
The label itself. The label, do you remember that specific length, specific label? The locations of those labels. So we know that sequence, and if we see a label in one position of those images, it means that that sequence appears in that specific region, location.
Okay.
Does it make sense? Okay.
I have some more questions. It doesn't interrupt it too much.
No, go ahead.
So on the same line of question, actually, but
sure.
How is distance measured? Is it? I guess it's not base pairs because you're not looking at base pairs. But is it centimeters? So
it's kind of base pairs starting with 0, but we cannot be exact. That's the problem. That's why it's floating point. Okay. So we have a molecule, right? We know it, and so we have relative position based on that. This is the 0. This is, however, the number of labels we have, and this is the relative position in it. Does it make sense? Yeah.
Then
it's kind of in base pairs because that's the metric, right? But we cannot be exact. That's why it's floating points.
Okay. Then, you said you linearized, you detect the molecule, label it, and at some point in the sequencer it gets linearized and it flows through nanopores essentially.
Nanochannel, I said.
Nanochannel.
Yeah.
It sounds an awful lot like nanopore. Why does this? It also sounded like it doesn't. Onur presented it in his introduction as it cuts the wool balls or the balls of DNA.
Mm-hmm.
Small
Yeah.
pieces.
Mm-hmm.
Is that happening here also? And if not, what's the magic sauce?
Yeah, it's happening, but it's not. So we have way larger molecules, so it's more intact than you would see in the sequencing world because of the wet lab operation. We are having extremely large molecules, sometimes even an arm length, chromosome arm length. So, and we don't have, we are not using electric current like nanopore is using, and we are using nanochannels to linearize the DNA. We are purely doing imaging. So we label fluorescently the DNA, and to linearize it, because it's like this, right?
Yeah.
And in order to linearize it, within the instrument, we are using nanochannels. And after it's linearized, so let me do this. So I have a video showing it. Maybe it will ease things a little bit. So is it sharing the full screen, John, or just the presentation? Is it directly showing this one, right? The desktop.
This is Sapphire
Okay.
from Bionano Genomics. With it, you can directly image the genome in high resolution to detect variations such as deletions, duplications,
I couldn't click on it.
translocations, and
Just a second.
inversions, known as structural variations,
Use
with
three
sensitivity
fingers.
as high as
Hmm?
99%. The
Just a
process is
sec.
simple and
John,
automated
can you
to
help?
enable routine use in disease research. Structural variation
It's messy.
commonly occurs in a broad range of diseases.
I'm trying to
including
make this big
neurodegenerative
screen, but the
diseases,
mouse is
intellectual
not really
disabilities,
working.
and
Okay.
various types of cancer.
Do you see it?
First, native ultra-high molecular weight DNA, multi-megabase pairs in
The
length,
video is
is
down
isolated.
there. It doesn't go down.
Fluorescent labels are enzymatically attached to these ultra-long molecules of DNA at a specific
There's an
sequence
issue with.
motif
Is there a
that
mouse?
occurs throughout the genome. The resulting label patterns are unique for each section of the genome. The full length of the DNA remains
Video is
intact
there.
during
Go up.
isolation and labeling,
Here. This one is the
preserving the
video.
architecture for the next step,
Okay.
imaging the genome. The labeled DNA is pipetted into the Sapphire chip and placed in the Sapphire instrument.
Okay.
In the chip, the DNA is uncoiled.
Okay.
This is Sapphire from Bionano Genomics. With it, you can directly image the genome in high resolution to detect variations such as deletions, duplications, translocations, and inversions, known as structural variations, with sensitivity as high as 99%. The process is simple and automated to enable routine use in disease research. Structural variation commonly occurs in a broad range of diseases, including neurodegenerative diseases, intellectual disabilities, and various types of cancer. First, native ultra-high molecular weight DNA, multi-megabase pairs in length, is isolated. Fluorescent labels are enzymatically attached to these ultra-long molecules of DNA at a specific sequence motif that occurs throughout the genome. The resulting label patterns are unique for each section of the genome. The full length of the DNA remains intact during isolation and labeling, preserving the architecture for the next step, imaging the genome. The labeled DNA is pipetted into the Sapphire chip and placed in the Sapphire instrument. In the chip, the DNA is uncoiled or linearized across thousands of parallel nanochannels and imaged, revealing its fluorescent label patterns. By capturing these images in repeated cycles across the hundreds of thousands of nanochannels on the chip, all the images necessary to assemble a map of an entire genome are captured. Algorithms extract molecules from the images, and then Bionano algorithms construct consensus genome maps. Bionano Access software also allows easy visualization and filtering of SV calls using our built-in control SV database. From the thousands of SVs found in any human sample, you can remove variants present in controls and narrow down your SVs of interest to a handful with just a few clicks. This is all enabled due to the extremely long molecules isolated and imaged with the Sapphire system.
Does it make sense now? Or at least makes more sense?
Still seems quite magic that these molecules stay longer than.
It's all about like the reagents, everything you are using. And as I said, we are not using nanochannels to measure or detect. We are just using it for linearizing them.
That's still quite a complex problem. I'm curious about how those channels work physically to be able to linearize such long pieces.
Yeah, it's whole, like the instrument itself. So yeah, the whole technology
But
itself.
Is
You
it
need to buy it to test
Is
it.
it inherent? So I think the key point for me is it inherent to this approach of optical mapping that they can stay that long, or can potentially, if let's say some unnamed other companies that do kind of nanopore based stuff, if they had a similar library preparation step and physical machine, could they do also the electrical signal-based sequencing of such long molecules?
So,
is it fundamental to optical imaging?
So because we are doing imaging, that's what affects our wet lab operation. So I don't know the details of how Nanopore, like ONT is, or other companies like that is performing their wet lab, or what makes it like less, like more chopped fragments. I don't know that. But what we are doing is labeling, like staining them. As far as I know, that's not Oxford Nanopore or something like that is doing. They are using nanopores to measure it. We are not using it to measure it. All of our workflow is based on imaging.
So yeah, I mean, this seems closer to like almost Illumina to some extent, right? Like okay, chemical and optical combination, fluorescent.
But the other technology also are doing labeling, which is
Yeah,
called
sure.
barcoding.
Yeah, sure.
So is the labeling here unique in terms of not ruining the molecule itself? That's why I'm maintaining
Possibly,
the
yeah.
sequence.
Yeah.
So there's no PCR replication.
No.
Okay.
So okay, and then so in the repeated cycle, so it's CCS kind of deal.
Not really, not really. So we are getting different molecules out of each imaging. So because the DNA is moving forwards, when it's moving forward, we are taking snapshots basically. So it's literally corresponding to other portions of the genome.
So because it says in the repeated cycles.
Yeah, the repeated cycles because it's moving, right?
Oh.
The DNA is moving within the channels.
I see. Okay.
Yeah, that's we are taking. It's like time shots, like time lapse. Does it make sense?
Honor is already looking at me. Yeah, stop interrupting.
Yes.
Okay.
I think you can leave the discussion.
Okay. Yeah. Let's discuss later.
Until after
Yeah.
Damla finishes her talk.
Okay.
Sounds good.
Okay. Yeah. All right. And
Otherwise, Jalkan may do something that you may not like.
yeah, you know what it is. Okay. So and with this workflow. Like OGM with Bionano can identify like all classes of structural variants, like the large ones. And what we do is we are basically observing the changes in spacing and comparisons of order and position of the labels basically and comparing them. That's how the analysis is taking place. So this is, again, because this is like an industry talk, the information is kind of like more high level. But so this is the pipeline, and I highlighted some portions of it. And this is the major stages of the analysis pipeline, and it's based on assembly. So it's of course includes alignment as well, but eventually the goal is to assemble the genome from the molecules. And the reason is that we are in clinical research and dealing with like revealing the structural variants. It means that it may not exist in the reference genome. We are trying to reveal those as well. So we cannot rely purely on alignment-based things. Instead, we are like constructing the genome from scratch. Or in the guided assembly scenario, using the reference as a reference, but not solely relying on that eventually because we assemble anyways. So when we look at the major stages, and this will give you a taste of the computation happening. So first we filter and sort the input molecules, basic, like nothing compute heavy there. Afterwards, optionally, we can estimate molecule error parameters. So
I'm trying to get rid of that so that we can see it, but
okay.
sorry.
No, that's fine. As long as I can do it.
Oh, okay, it's a different screen.
Yeah.
But I would do it easily.
Yeah, okay. It says de novo and guided assembly pipelines with OGM data in the title.
Okay.
So, yeah. Yeah.
Trust you.
Yeah. So the next stages after filtering the molecules and sorting them is trying to estimate the error parameters. What we mean by error parameters is, for example, the label like distances between them could be rescaled because there might be some errors about the estimation out there, or the positional like information about the labels. That's the second step. And then depending on whether we do this de novo or guided using a reference, the path is kind of diverges, as you can imagine. So if it is de novo, it means we don't have any reference or we don't want to use it. In that scenario, similar to overlap layout consensus that we know from sequencing to perform assembly, what we do is perform pairwise alignment and use that between the molecules and use that information to construct a draft genome. So we call them draft consensus maps. And if we want to use a reference, then we don't do these two steps. We use the reference maps as our starting draft consensus maps, and then they start like the stages merge again at that point. And what we do, we have a draft map, we have a set of molecules. What we can do, we align those molecules to those draft maps, and then we refine the maps. Basically, by using that alignment information, we improve the quality of the consensus maps. And then the next step is, after we have one set of refinement, the maps could change, right? And let's say we have this map, and maybe they have an overlap between them, so they can be merged. That's what extend and merge stages are doing. We have several iterations of those, so we map pairwise map the consensus maps, or that's how we do the merging step. And for extend, we use the molecules, map the molecules to the edges of the draft, like the consensus maps, try to lengthen them, and after lengthening them, trying to merge several consensus maps together. So the goal is to make them larger and larger so that we have better coverage. And that's step 6, which happens multiple times. And then we have one final refinement stage with the extended and merge consensus maps. And that after doing that, again, map the molecules to the consensus maps, improve them again, and that makes our final set of maps, consensus maps. And we use those consensus maps against a set of reference maps to detect the SVs, basically what is different within this generated set of maps, basically. So this is very high level, I know. But why I highlighted summers, because you can imagine we have an idea about the sequencing world, what is computationally expensive. So alignment, refinement, all these things which involve operations are expensive in the context of OGM analysis as well. Which makes it even worse is, one, the floating point operations. We are not dealing with ACGTs, it is not that way. And the flavor of the algorithm is very similar to what we see in Smith-Waterman, but it has a way larger, deeper recurrence. And the innermost loop is extremely heavy with a lot of floating point-based operations. So that's what's making this problem different than sequencing, and to be honest, much more complex and expensive.
Do you have a name for the algorithm?
It's called RefAligner, but as you can imagine, it's not open source. So we have the binary out there, I guess, but no, it should be, because whatever we are shipping in terms of compute solution, the binary is there, but it's not reverse engineerable. So
Time warping ancestors don't work here.
huh?
The dynamic time warping algorithms and such.
No. Any questions? Yeah.
Actually, I'm not sure if I understand correctly, but from my understanding, especially on steps 6 and 7, you consistently use previous outputs to generate newer inputs.
Correct.
So I feel like this would also multiply the amount of errors you've had in the previous steps. So what do you do to prevent that?
So basically you are trying to eliminate them with that. So you are trying to get rid of, because you have coverage, right? You don't, similar idea, similar explanation what we have in sequencing. So multiple molecules per region. So by using that information, we are trying to basically co-merge. And at one point the maps don't change anymore. And that's the point where you say, okay, this is what we have. So does it make sense? Okay. Any other question? Okay. So this is where we are right now. And where we are going is what is motivating what I'm doing at Bionano, basically. So these are 4 like evolution points that we as Bionano are like aiming towards. And what is relevant here is the first and third things. And third thing is what I'm doing basically, but it's related with the first one, which is enhancing throughput. So it's a public information. We are like working on a new like Sapphire instrument, which is high throughput. And when I say, and I kind of glimpsed over this in my first talk as well, like genomics companies are improving their instruments a lot. And as part of that, in Bionano, we are also coming up with a new instrument, which is extremely higher throughput than the current Sapphire system. And as you can all imagine, that comes up. So even in the current analysis pipeline, we are dealing with 16 hours, 24 hours per sample pipeline time, which is a lot. So, and with this, the throughput will be increasing significantly, so it is impossible to keep up with that. So this is very like familiar to all of us. We knew this, and this is the motivation for what I'm doing at Bionano, because we need to improve our analysis pipeline in some way so that it can keep up with this, not just this new machine, but the existing one is like we are releasing new software, we are releasing new reagents, we are upgrading our software. Even with that, the existing instrument is getting better as well. But analysis pipeline needs to keep up with that. And that's what motivates my work at Bionano. And that's the hardware acceleration part in my title. So, and it's a common goal for all genomics companies, as we have to decrease the cost. And the goal is that, the reason is so that a lot of people can access what we are providing here in the rural areas in everywhere, because these machines are not cheap. So, so the number of samples you are getting out of it, and the analysis time, analysis cost, all those kind of things build upon that. So we have to decrease everything, not just the instrument. And that's the goal of my work, which is the, I don't know why it's not visible, but the next generation compute solution we are working on. And the goal is here is, as you can imagine, significantly reducing the time and cost per like sample. And with that, what we are marching towards is decreasing the pipeline processing time end to end by roughly 8 times, which will drop it roughly to 2 hours per sample, so that it can kind of keep up with the high throughput system. And to give you some overview of how it looks like, so this is the instrument. It has a controller next to it, which is controlling the instrument basically and gathering the data. In the current system, we have a cluster of servers which is sitting in the customer's IT rooms, which is causing a lot of issues, as you can imagine. So the goal in this project is not just providing like better cost, like better throughput, but also like removing that burden on the customer. So that's why we are coming up with a workstation kind of single machine that can scale in the future if needed, but can sit next to the instrument, which is coming up with another constraint, because it will sit next to the instrument, next to everyone like working on the lab. So it should be efficient enough in terms of power, noise, all those kind of things that you can imagine. And why it is like, as you can imagine, again, because of the, so we tested several different hardware accelerators, and we revealed that NVIDIA GPUs are the best fit for our current solutions. And we are working on a CUDA optimized pipeline that is specifically working on those stages I highlighted: alignment, refinement, and SV detection. And I kind of like highlighted why NVIDIA GPUs while I was explaining the pipeline. But to be specific, we are also using the new generation of NVIDIA's RTX 6000 ADA generation cards for this work. So to summarize, with this new solution, we are aiming to accelerate the analysis for fast time to results. And as we can imagine, this is the main goal is to support higher throughput. And again, this is, we can call it as hardware, software, like co-design approach, because yes, it is GPU mapping, but we all know that we cannot directly map, so it's a completely different hardware. So that's requires new algorithms or new approaches from our side as well. And the goal, our goal is to drop the highly complex cancer analysis less than 2 hours so that we can keep up with the instrument. And again, we are aiming to have a workflow that can sit in a lab environment. So for this specific project to work with me, we have an opening, like job opening, specifically for GPU engineers right now. If anyone is interested in, you can talk with me. And so we presented this as part of a workshop recently. So it's a very similar presentation for my side, but for some more inspiration on how like this is really used in the clinical space, I would recommend looking at that as well. Thank you.
Thanks, Damla.
Thank you.
Any questions?
Too many.
Yeah, now that we have no more break.
Well, you cancel the break.
Yeah, 6 more talks.
So what makes it unique to SV detection?
So several things. One thing is, as I mentioned, we are trying to have an end-to-end workflow that has very limited like manpower need, which is not there yet. Like in the outside, in the status quo basically approaches, there's a lot of human involvement. We are trying to replace that and have an automated end-to-end workflow. That's one. The second one is I show you the spectrum. The resolution that we are providing is pretty huge. So that's another big perk, basically.
But is that makes it strictly for SV detection, or it can support other applications regardless if it is endow, SNPs?
So SNPs and small indels are not supported because of the limitations of the technology. That's why like company
coarse grain within the genome.
Yeah. So, but more strong, like larger, like higher than 500 pp, as I showed in the spectrum. That's the target space. That's why, as I also showed, it's not a replacement for sequencing or NGS. It's a complement for that. So, OGM, yeah.
And none of the analysis tools are open source?
No.
None?
None.
Not even development version?
No.
Okay.
Is
No.
there data available in any way?
There are available data, yes.
Yeah, but if you don't have analysis, then you cannot do much.
You can do analysis, but you cannot reveal much from the.
But we never
but-
can compete with
but-
the original tool.
But I am not sure about this. But one thing is, while doing my work, in order to get support from several different hardware companies, we signed NDAs, work with them. So it's always a possibility, which I also want to push, maybe.
Okay,
Yeah.
So you are open for that?
I am. We can always bring it. Yeah. But I'm someone coming from academia. I really believe the connections with academia. I really believe in that, and I'm trying to vocalize that as well. But we cannot know without trying. So
Sounds good.
yeah, that's why I'm here.
Thank you.
Okay. I think the rest can be taken offline.
Sure.
Thanks, Damla.
Thank you.
Nice overview of what you're doing.
Yep.